The call came into the Marine Research Station on the southwestern coast of Sri Lanka at 4 in the morning, and the scientist who answered it has described the next 40 seconds of that conversation as the most disorienting professional experience of her career, which spans 23 years of deep-sea biological research across 3 ocean basins and includes 2 first-hand encounters with species that were, at the time of their discovery, unknown to science. The call was from the captain of a commercial fishing vessel operating approximately 340 kilometers southwest of the Sri Lankan coast, in a section of the Indian Ocean that sits above one of the deepest and least explored sections of the global ocean floor, a region where the seafloor drops below 5,000 meters across an area larger than Western Europe, and where the biological communities living at those depths have been sampled by fewer scientific expeditions than have visited the surface of the moon. The captain was not calling to report a catch. He was calling because something had come to the surface alongside his vessel that he did not have a category for, and that his crew of 11 experienced fishermen, men who between them had logged more than 200 collective years on the Indian Ocean, had not seen before and were not prepared to describe in terms that corresponded to anything in the existing vocabulary of things that live in the sea. The object, and the captain used that word initially before correcting himself and using the word creature, and then stopping and saying he was not certain which word was more accurate, was large. Not large in the way that whale sharks are large, familiar and proportioned and recognizable as a living thing of a specific type. Large in the way that something is large when its scale does not immediately register as biological. When the brain reaches for the category of structure or vessel or geological feature before it accepts that what it is looking at is alive. It was at the surface. It was not moving in the way that distressed or dying animals move, not thrashing, not listing, not showing the behavioral signs of an organism that has ascended beyond its pressure tolerance and is experiencing the physiological consequences of rapid decompression. It was present at the surface in the pre-dawn darkness of the southwestern Indian Ocean, and the fishing vessel's floodlights were illuminating something that the captain, after 40 seconds of attempting to describe it to the marine researcher on the other end of the phone, summarized in a sentence that she wrote down immediately and that has been shared widely in the deep-sea biology community in the weeks since. It looks like the ocean decided to turn itself inside out and show us something it was keeping. That sentence is not a scientific description. It is the attempt of a man with 2 decades of ocean experience to communicate in the only language available to him at 4 in the morning with a ship full of frightened crew and something enormous at the surface alongside him, what he was seeing. The marine researcher who took the call did not dismiss it. She had worked with this captain before, on a citizen science program that recruited commercial fishermen to report unusual sightings and collect biological samples from their operations. And she knew him as a precise and unsensational observer who did not call research stations at 4 in the morning unless the situation warranted it. She asked him to activate the vessel's onboard camera system, which had been installed as part of the citizen science program and which transmitted footage via satellite to the research station's servers. The footage began arriving in fragments, compressed by the satellite uplink into low-resolution video that nonetheless showed enough to cause the researcher to wake 3 colleagues and place 2 more phone calls before the sun rose over Sri Lanka. What the footage showed has not been publicly released in its full form. Portions of it have been described by the researchers who have seen it in terms that are careful and qualified, and that consistently emphasize the preliminary nature of any interpretation, but that consistently fail to produce a description that corresponds to any known species in the deep-sea biological literature. The Indian Ocean, which is the 3rd largest ocean on Earth and the warmest, covers approximately 70 million square kilometers and reaches its greatest depths in the Java Trench, which extends to nearly 7,300 meters, and in the lesser-known but equally profound deep basins of the central and western Indian Ocean, including the Somali Basin, the Arabian Basin, the Mid-Indian Basin, and the Crozet Basin, each of which descends below 5,000 meters, across areas that have been visited by deep-sea research expeditions on so few occasions that the biological communities living there are essentially uncharacterized by modern science. The fishing vessel was operating above the northern margin of the Mid-Indian Basin in water approximately 4,800 meters deep when the sighting occurred. The depth is significant not as a measure of distance, but as a measure of the biological and physical conditions at the seafloor below, conditions that define what kinds of organisms are capable of living there and what it would mean, biologically and physically, for something from that depth to be at the surface. That meaning is the spine of this story, and working through it carefully, with honesty about what is known and what is not, is the only way to understand why marine biologists who have spent their careers studying the deep ocean and who are not given to dramatic language are using the word terrified. The biology of deep ascent is one of the most important and least publicly understood fields in marine science, and understanding it is essential to understanding why the sighting off the Sri Lankan coast has produced the response it has produced among the researchers who have been briefed on the footage and the associated data. Every organism that lives in the deep ocean is adapted to the specific pressure, temperature, chemical, and light conditions of its depth zone, and those adaptations are not superficial. They are not the biological equivalent of wearing a coat in cold weather, a behavioral or physiological adjustment that can be reversed when conditions change. They are fundamental molecular-level modifications to the biochemistry and cellular structure of the organism that make life possible at depth and that simultaneously make life at the surface impossible without a transition period that, for most deep-sea organisms, does not exist in any survivable form. The pressure adaptations of deep-sea organisms illustrate this most clearly. At 4,800 meters depth, the pressure is approximately 480 atmospheres, meaning every square centimeter of an organism's body is subjected to a force equivalent to the weight of 480 kilograms. The proteins and lipids that constitute the biological machinery of every living cell are sensitive to pressure. At the pressures of the deep ocean, the proteins of surface-dwelling organisms would denature, losing their three-dimensional structure and their biological function in the same way that the proteins in an egg white denature when you heat them. Deep-sea organisms avoid this by having proteins with amino acid sequences that maintain their functional structure under high pressure, and cell membranes with lipid compositions that remain fluid rather than solidifying under the conditions of the deep. These molecular adaptations are the product of millions of years of evolutionary selection in the specific pressure environment of the deep ocean, and they are as specific and as non-transferable as any other evolutionary adaptation. An organism adapted to 480 atmospheres of pressure does not simply tolerate reduced pressure when it ascends toward the surface. Its proteins, optimized for high-pressure function, begin to malfunction at low pressure. Its cell membranes, optimized for high-pressure fluidity, become excessively fluid at low pressure, losing their selective permeability and their ability to maintain the chemical gradients that drive cellular metabolism. Its gas-filled spaces, if it has any, expand as pressure decreases, potentially to the point of rupturing the tissues that contain them. The ascent from 4,800 meters to the surface, if it occurs rapidly, is not an adventure. It is a physiological catastrophe, and the organisms that survive it in any functional sense are those that either have no gas-filled spaces and whose biochemistry is robust enough to tolerate the transition, or those that have evolved specific mechanisms for managing the pressure change during ascent, mechanisms that are known in some migratory deep-sea species, but that are rare and highly specialized. The fact that the object described by the fishing vessel captain appeared at the surface and was not displaying the behavioral signs of a dying organism is therefore not a reassuring detail. It is an anomalous one. It is the detail that caused the marine researcher who took the captain's call to wake her colleagues before sunrise, because an organism of apparent deep-sea origin that is present at the surface and is not in obvious physiological distress is not something that the existing biological framework for deep-sea organisms easily accommodates. The deep scattering layer, that community of mesopelagic organisms that performs the daily vertical migration between roughly 200 and 1,000 meters depth, demonstrates that vertical movement in the ocean is biologically possible and biologically important. The organisms of the deep scattering layer are adapted for migration, with biochemistries and body plans that can tolerate the pressure and temperature changes of their daily transit. But they migrate within a specific depth range that is far shallower than the 4,800 meters at which the fishing vessel was operating. And the physiological demands of migrating between 200 and 1,000 meters are categorically different from the demands of ascending from 4,800 meters to the surface. The mesopelagic migrants are the commuters of the ocean, traveling a known route between familiar environments on a daily schedule. An organism ascending from 4,800 meters to the surface is doing something different in kind, not just in degree, and the biological mechanisms required to do it without apparent distress are mechanisms that have not been documented in any known deep-sea species from the Indian Ocean abyssal zone. This is the first layer of why the marine biologists who have seen the footage are using the word terrified. Not because what they saw is obviously dangerous, but because what they saw is obviously impossible under the frameworks they have spent their careers building, and impossible things in science are not comfortable. They are demanding. They require the revision of assumptions that entire research programs are built on, and the revision of those assumptions has consequences for everything downstream of them, including the understanding of what the deep ocean contains and what it is capable of producing at the surface under conditions that are, as this story will establish, not stable. The temperature adaptations of deep-sea organisms add another layer to the ascent problem. At 4,800 meters in the Indian Ocean, the water temperature is between 1 and 3 degrees Celsius, cold enough to slow enzymatic reactions to the point where they would be lethally inefficient in a surface-adapted organism. Deep-sea organisms compensate with enzymes that are optimized for low-temperature function, with higher flexibility in their active sites and lower activation energies that allow them to catalyze biological reactions at rates compatible with life in cold water. Those same enzymes, exposed to the 28 to 30 degree surface temperatures of the tropical Indian Ocean, would operate too fast, accelerating metabolic reactions beyond the organism's capacity to manage their products and consuming energy stores at rates that quickly become unsustainable. The thermal shock of ascending from 2 degrees to 28 degrees in the time it takes a buoyant object to rise through 4,800 meters of water would be lethal for virtually any known deep-sea organism, because the temperature change is too large and too fast for any known biological thermoregulatory mechanism to manage. And yet the footage shows something at the surface that is not dead, something that the captain described as present in a way that implies behavioral agency rather than passive floating, that the floodlights of the fishing vessel illuminated for approximately 40 minutes before it submerged again, not sinking in the way that a dead or dying object sinks, but descending in the way that a living thing descends when it chooses to, with a directionality and a control that the researchers who have reviewed the footage have described, carefully and with appropriate qualification, as volitional. a volitional descent from the surface of the tropical Indian Ocean back toward 4,800 meters of cold, dark water by something that should not have been able to survive the ascent in the first place. That is what the footage shows, and that is why the word terrified is not an overstatement. The footage from the fishing vessel's onboard camera system arrived at the Sri Lankan research station in 17 separate compressed video fragments over a period of approximately 90 minutes. The transmission interrupted twice by the satellite uplink limitations of a commercial fishing vessel operating at the edge of its connectivity range in the pre-dawn hours of the southwestern Indian Ocean. The researcher who received the fragments assembled them in sequence and watched the composite footage 4 times before calling her first colleague, and the detail that she returned to on each viewing, the detail that she described in her notes from that morning as the thing that made the footage impossible to dismiss as a misidentification or an optical artifact, was not the size of the object, though the size was remarkable, and not the apparent behavioral agency of its descent, though that too was remarkable. It was the light. The object at the surface of the Indian Ocean was producing its own light, and the light it was producing was not bioluminescence in the form that marine biologists are accustomed to seeing in deep-sea organisms. Bioluminescence is one of the most widespread and most studied phenomena in ocean biology. It is produced by the majority of deep-sea organisms, from bacteria to fish to squid to jellyfish, through a chemical reaction involving a light-emitting molecule called luciferin and an enzyme called luciferase, and it serves a variety of biological functions including predator avoidance, prey attraction, communication, and camouflage through counter-illumination. The bioluminescence of deep-sea organisms is typically blue or blue-green in color, a characteristic that reflects the spectral properties of the luciferin-luciferase reaction, and that is evolutionarily optimized for the optical properties of deep seawater, which transmits blue wavelengths more efficiently than other parts of the visible spectrum. It is typically dim, visible to the dark-adapted eye, but not overwhelming, and it typically appears in specific patterns, flashes, pulses, glowing patches, or lines of light that correspond to the anatomical distribution of the light-producing organs called photophores. What the footage from the fishing vessel shows is different from all of these characteristics in ways that the researchers who have analyzed it are finding difficult to explain within the existing framework of bioluminescent biology. The light produced by the object at the surface is not blue-green. It is described by the researchers as white with a yellowish cast in its central regions and a blue-green periphery, a spectral distribution that does not correspond to any known bioluminescent chemistry in the marine literature. It is not dim. It is bright enough to be visible against the background illumination of the vessel's floodlights, which means its intensity is orders of magnitude above the typical output of known bioluminescent organisms. And it is not patterned in the way that photophore distributions are patterned. It appears to emanate from the entire surface of the object rather than from discrete anatomical structures, a diffuse, whole-body illumination that has no parallel in the documented biology of any known marine species. The researchers who have reviewed the footage have considered and evaluated several alternative explanations for the light. Reflection of the vessel's floodlights is the most obvious alternative, but the footage shows light emanating from portions of the object that are not oriented toward the floodlights and that are partially obscured from them by the object's own mass, making passive reflection an inadequate explanation for the full spatial distribution of the observed illumination. Phosphorescence, the emission of light by material that has previously absorbed radiation, is another possibility, but phosphorescent emission in marine contexts is typically associated with specific dinoflagellate species that produce a blue flash when mechanically disturbed, and the sustained, whole-body white-yellow illumination in the footage does not match the characteristics of dinoflagellate phosphorescence. Chemiluminescence from a chemical reaction other than the standard luciferin-luciferase system is a theoretical possibility, but the known chemiluminescent reactions in marine organisms all fall within the blue-green spectral range of standard bioluminescence, and a white-yellow chemiluminescent system in a marine organism would represent a biochemical novelty with no known precedent. The light, in short, is anomalous by the standards of every known mechanism of biological light production in marine organisms, and it is one of the details that elevates the footage from interesting to genuinely scientifically significant, because it implies not just an organism of unknown species, but an organism with a biochemical capability that does not exist in the current catalog of marine biological chemistry. Before this goes further, and because what follows is going to require the kind of sustained attention that this kind of story deserves, this is the moment to subscribe if you have not already. What the researchers found when they began cross-referencing the footage with the historical record of Indian Ocean deep-sea research is one of the most unsettling convergences of data points that the deep-sea biology community has encountered in recent decades. And the cross-referencing is still ongoing, and every thread they pull leads to another thread. And the picture that is assembling itself from those threads is the reason that 3 separate institutions have now requested access to the original footage, and that 2 of those requests have been declined pending a review process whose timeline and institutional oversight have not been disclosed. Subscribe, because this story is moving fast in the scientific community, and this channel will be here at every development. The size estimation from the footage is another element that the researchers have spent significant time analyzing, because size is one of the few parameters that can be estimated with reasonable confidence from video footage when a reference object of known dimensions is in the frame. The fishing vessel itself is the reference object, and its dimensions are known from the vessel's registration documents. The object at the surface, estimated by comparison with the vessel's hull length and superstructure height in the footage, is approximately 20 to 28 meters in its longest visible dimension, with the uncertainty range reflecting the difficulty of estimating the full extent of an object whose boundaries are not always clearly defined against the dark water surface. 20 to 28 meters. For comparison, the largest known cephalopod, the colossal squid, reaches a maximum documented length of approximately 14 meters, and that record is based on a single specimen. The giant squid, more commonly encountered and better documented, typically reaches 10 to 13 meters. The largest known fish, the whale shark, reaches approximately 18 meters at the extreme upper end of documented sizes. The object in the footage, at its estimated size, exceeds the documented maximum size of every known marine invertebrate and approaches, or exceeds the size of the largest known fish. If it is a single organism rather than an aggregate, such as a colonial siphonophore or a large aggregation of smaller organisms behaving collectively, its size places it in a category that has no established representative in the Indian Ocean deep-sea fauna as currently documented. The colonial organism possibility is one that the researchers have taken seriously, because colonial organisms, particularly siphonophores of the class Physonectae, are known to achieve extraordinary lengths through the aggregation of individual zooids into a functionally integrated colonial body, and the deep Indian Ocean siphonophore fauna is very poorly documented. A large siphonophore colony at the surface would not, under normal circumstances, produce the behavioral characteristics described in the footage, specifically the volitional descent, because siphonophores are generally at the mercy of currents and buoyancy rather than capable of directed swimming. But the deep-sea siphonophore fauna includes species with pneumatophores, gas-filled floats, that provide some degree of buoyancy control and a colonial organism with sufficient pneumatophore capacity and sufficient nectophore, swimming bell, coordination could, in principle, achieve the kind of directed vertical movement described in the footage. The colonial organism hypothesis does not resolve the bioluminescence anomaly because the light characteristics in the footage do not match those of any known siphonophore species, but it provides a framework within which the size and some of the behavioral characteristics could be accommodated without requiring the existence of a single organism of unprecedented dimensions. The researchers are not settled on either interpretation. The footage is being analyzed with every tool available, including frame-by-frame intensity mapping of the bioluminescent emission, geometric reconstruction of the object's three-dimensional form from the two-dimensional video record, and spectral analysis of the light using the color channels of the camera system as a crude but informative spectrometer. Each analysis adds constraints to the set of possible explanations, and each constraint, rather than narrowing the field toward. A single answer has so far been most effective at eliminating explanations that initially seemed plausible and leaving behind a smaller set of possibilities that are progressively more difficult to accommodate within the existing biological framework of Indian Ocean deep-sea life. The historical record of anomalous deep-sea sightings in the Indian Ocean is longer and more substantive than the public record suggests, and cross-referencing the fishing vessel footage against that historical record is one of the analytical threads that the research team assembled around the Sri Lankan sighting has been pulling with the most focused attention, because the historical record contains details that, in the context of the current footage, have acquired a significance they did not appear to have when they were first reported. The Indian Ocean is the least studied of the three major ocean basins. It has received fewer deep-sea research expeditions than either the Pacific or the Atlantic. Its deep biological communities are documented at lower resolution than equivalent communities in either of the other two basins, and its history of anomalous sighting reports from commercial fishing fleets and merchant shipping has been systematically collected and analyzed less thoroughly than the equivalent records for the Pacific and Atlantic. This is partly a function of geography. The Indian Ocean is bordered primarily by developing nations with limited marine research infrastructure, and the international research programs that have conducted deep-sea expeditions in the basin have concentrated their effort in the biologically and geologically distinctive regions, the mid-ocean ridge system, the hydrothermal vent fields of the central Indian Ridge, and the extreme depths of the Java Trench, leaving the vast stretches of the central and western abyssal basins essentially unvisited by modern deep-sea research. The historical record that does exist for anomalous sightings in the Indian Ocean abyssal zone goes back further than most people realize, and it is more internally consistent than a collection of anecdotal reports from non-scientists might be expected to be. The consistency is what makes it significant, because the reports come from different times, different locations, different observers with different cultural backgrounds and different vocabularies for describing what they encountered, and they share a set of characteristics that suggests they may be describing encounters with the same phenomenon or the same category of phenomenon rather than being independent fabrications or misidentifications. The earliest reports in the systematic record that the research team has been able to locate date from the mid-19th century, from the logs of British merchant vessels operating on the India to Australia trade routes that crossed the central Indian Ocean above the mid-Indian basin. The log entries are brief and written in the restrained language of professional mariners who were disinclined to make claims that would invite skepticism from their employers. But several of them describe encounters with large, luminous objects at or near the surface that were observed for periods of minutes to hours before submerging that did not match the appearance or behavior of any known marine animal, and that the reporting officers noted with apparent discomfort as something they had no adequate description for. The geographic coordinates of these historical reports, extracted from the log entries and plotted on modern bathymetric charts, cluster in the northern and central portions of the mid-Indian Basin, in water depths between 4,500 and 5,200 meters, an area that overlaps substantially with the location of the current fishing vessel sighting. The clustering is not conclusive. Historical navigation was imprecise by modern standards, and the coordinate uncertainty in 19th century log entries is large enough that individual reports cannot be confidently localized to within tens of kilometers. But the clustering across multiple independent reports from multiple decades, all converging on the same general section of the Indian Ocean above the same abyssal basin, is the kind of pattern that deserves more than dismissal as coincidence, particularly when the pattern persists into the 20th century. The 20th century record contains several reports from the crews of research vessels that were operating in the Indian Ocean on geological and physical oceanographic surveys, reports that were logged internally but not published, in some cases because the scientists involved lacked the biological expertise to evaluate what their crew members described, and in other cases because the sighting occurred outside of working hours, in darkness, and was not accompanied by any instrumental data that would provide an evidentiary foundation for a published report. One of these unpublished reports, from a German oceanographic survey vessel operating in the mid-Indian Basin in 1971, describes an encounter that is remarkably similar in its key characteristics to the current fishing vessel sighting: a large, luminous object at the surface, observed for approximately 30 minutes before descending, estimated by comparison with the vessel's hull to be in the range of 15 to 20 meters in its longest dimension, producing light that the reporting officer described as white rather than the blue-green of typical ocean bioluminescence, and showing no signs of distress despite its apparent deep-sea origin. The 1971 report was filed in the ship's log and in the expedition's internal documentation, and it was apparently shared informally within the German oceanographic community at the time. But it was never published, and it appears to have been filed and forgotten, as such reports tend to be when they arrive without instrumental corroboration and without an obvious framework for their interpretation. The research team that is currently analyzing the Sri Lankan footage located the 1971 report through a search of German oceanographic archive records, conducted after a retired researcher who had been a junior scientist on the 1971 expedition read a description of the current sighting in an informal scientific newsletter and contacted the Sri Lankan research station to report that he had seen something similar 45 years earlier. That contact, a single email from an 80-year-old retired oceanographer, opened an archival thread that has since produced 4 additional historical reports from the 20th century, each independently filed, each describing encounters with large, luminous objects in the mid-Indian Basin, each sharing the key characteristics of the current sighting, and each having been stored in institutional archives without being connected to the others, because nobody had previously had a reason to search for them simultaneously. 4 additional reports, from different decades, different vessels, different nationalities, different institutional contexts, all describing the same things: large, luminous, deep origin, surface present, voluntarily descending. The convergence of the historical record with the current footage is the finding that has most significantly elevated the assessment of the sighting among the researchers involved, because it transforms the current event from a potentially unique anomaly into the most recent instance of a recurrent phenomenon that has apparently been occurring in the mid-Indian Basin for at least 150 years without attracting the sustained scientific attention required to characterize it. A phenomenon that has been occurring for 150 years without characterization is not a new discovery. It is an old oversight, and the scale of the oversight is itself informative because it tells you something about the depth of the Indian Ocean's biological darkness. about how thoroughly the absence of monitoring infrastructure and scientific attention can hide a phenomenon of apparent size and regularity from the community that should be documenting it. The research team is now in the process of extending the archival search beyond the German and British records that have so far been the primary sources, reaching into the records of French, Indian, Soviet, and Japanese oceanographic expeditions that operated in the Indian Ocean during the 20th century, and into the logs of the commercial shipping fleets of multiple nations that have crossed the mid-Indian basin on regular routes for more than a century. The search is slow because the relevant archives are distributed across multiple countries and multiple institutions, many of them not digitized, some of them in languages that require translation, and all of them requiring the kind of targeted manual search that cannot be automated because the relevant entries are embedded in documents whose primary purpose was something entirely different. But the preliminary results of the search, the 5 historical reports already located, and the pattern they establish, are sufficient to justify the effort. And the researchers conducting the search are doing so with the focused attention of people who believe that the archive contains more of what they are looking for, and that what they find will matter to the interpretation of what the fishing vessel camera recorded at 4 in the morning in the southwestern Indian Ocean. The phenomenon, whatever it is, has a history. The history has been hiding in plain sight in the archives of institutions that did not know they were holding it. And the current footage, arriving in 17 compressed video fragments over a satellite uplink in the pre-dawn hours of a Tuesday morning, is the event that finally provided the reason to look. The Indian Ocean's physical oceanography provides a set of environmental conditions that are, in several important respects, distinct from those of the Pacific and Atlantic basins, and those distinctions are not incidental to the story of what has been observed in the mid-Indian basin. They are potentially explanatory in the sense that they describe a set of physical and chemical conditions in the deep Indian Ocean that differ from the conditions in the better-studied deep basins of the other 2 major oceans in ways that could support biological phenomena that have not been documented elsewhere. Understanding those distinctions requires spending time with the physical oceanography of the Indian Ocean in enough detail to see why the deep Indian Ocean is not simply a warmer, smaller version of the deep Pacific or Atlantic, but a system with its own circulation dynamics, its own water mass structure, its own chemical regime, and its own biological context that has been shaped by millions of years of geological and oceanographic history that has no exact parallel in any other ocean basin. The Indian Ocean is unique among the major oceans in having no connection to the Arctic. The Pacific connects to the Arctic through the Bering Strait, and the Atlantic connects through the passages between Greenland, Iceland, and the British Isles. And those connections allow cold, dense, oxygen-rich water formed at high latitudes to sink and spread along the ocean floor, ventilating the deep basins of both oceans with relatively young, oxygen-rich bottom water. The Indian Ocean has no northern high-latitude source of dense bottom water because its northern boundary is the Asian continent, and the deep water that fills its abyssal basins is derived entirely from the Southern Ocean, entering through the deep passages south of the basin and spreading northward along the seafloor in a slow, deep circulation that is less vigorous and less well ventilated than the deep circulation of the Atlantic or Pacific. The consequence of this circulation difference is that the deep Indian Ocean is older and less oxygenated than the deep Pacific or Atlantic at equivalent depths. Older in the oceanographic sense means that the water has been out of contact with the atmosphere for longer, and less oxygenated means that the dissolved oxygen that was present when the water sank has been progressively consumed by the microbial respiration of sinking organic material over the centuries since the water last saw the surface. The oxygen minimum zone of the Indian Ocean is one of the most severe in the global ocean, particularly in the northern Indian Ocean, where the restricted circulation of the Arabian Sea produces bottom water oxygen concentrations that are among the lowest recorded anywhere in the open ocean. These low oxygen conditions extend deeper into the Indian Ocean water column than in the Pacific or Atlantic, and the deep basins of the central and western Indian Ocean, including the mid-Indian Basin, have deep water oxygen concentrations that are lower than equivalent depths in other ocean basins. Low oxygen conditions in the deep ocean have profound biological consequences. They select strongly against aerobic organisms, which require dissolved oxygen for their metabolism, and they favor the anaerobic and microaerophilic organisms that can function in near-anoxic environments. They alter the chemistry of the sediment-water interface, shifting from oxidizing to reducing conditions that change the cycling of metals, sulfur, nitrogen, and carbon in ways that support different microbial communities and different chemical reactions than those operating in well-oxygenated deep-sea environments. And they create a distinct selective pressure on any organisms that are motile enough to move between different depth zones, favoring those with physiological adaptations for tolerating hypoxic conditions during their vertical migrations. The biological community of the deep Indian Ocean, shaped by millions of years of evolution in this low-oxygen, warm, poorly ventilated environment, may therefore differ from the deep Pacific and Atlantic communities in ways that go beyond the taxonomic differences expected from geographic isolation alone. It may contain organisms with physiological adaptations to hypoxic conditions that have no parallel in the better oxygenated deep basins, organisms whose biochemistry has been optimized for survival in conditions that would be stressful for deep-sea organisms from other basins. Those adaptations, if they include enhanced tolerance for the chemical and thermal changes associated with vertical ascent through the Indian Ocean water column, could provide a partial explanation for an organism's capacity to reach the surface. From abyssal depths, without the physiological catastrophe that would be predicted for a deep-sea organism from a better oxygenated basin. Partial is the operative word. Enhanced hypoxia tolerance does not resolve the pressure adaptation problem, does not resolve the temperature shock problem, and does not explain the anomalous bioluminescence. But it contributes to a picture of the deep Indian Ocean as a biological environment that may have produced adaptations not present in the deep sea fauna of other basins. And that picture is relevant to assessing how unusual an organism with the characteristics described in the fishing vessel footage would need to be relative to the known Indian Ocean deep sea fauna, as opposed to the deep sea fauna of other basins. The thermal structure of the Indian Ocean adds another dimension to this picture. The Indian Ocean is the warmest of the major ocean basins at the surface, with tropical surface temperatures regularly exceeding 28 degrees Celsius across the central and northern basin, and the warmth penetrates deeper into the water column than in the Pacific or Atlantic because the weaker deep circulation of the Indian Ocean brings less cold bottom water upward to cool the intermediate layers. The thermocline of the Indian Ocean, the depth zone where temperature drops most rapidly with increasing depth, sits at a shallower depth and spans a narrower depth range than in the other major basins, and the temperature gradient through the upper water column is steeper. For an organism ascending from abyssal depths, the steeper temperature gradient means that the thermal shock of ascent is concentrated in a shallower depth range, which could in principle be navigated more quickly, and the warmer intermediate water temperatures mean that the organism encounters a less extreme thermal environment in the mid-water column during its ascent than it would in the colder Pacific or Atlantic. These are not explanations. They are physical context that makes the thermal aspects of the ascent problem slightly less absolutely prohibitive in the Indian Ocean than in other basins, and that context contributes to the assessment of whether an organism capable of ascending from abyssal depths to the surface could plausibly exist in the Indian Ocean deep-sea fauna. The chemical environment of the deep Indian Ocean provides one more contextual piece that the researchers have been examining in relation to the current sighting. The mid-Indian Basin, where the fishing vessel made its observation, sits above a section of the Indian Ocean seafloor that is associated with a distinctive geochemical province characterized by elevated concentrations of certain dissolved metals, particularly manganese and iron, in the bottom water, and by sediment chemistry indicative of enhanced hydrothermal fluid input from the mid-ocean ridge system that runs through the central Indian Ocean. Hydrothermal fluids from the central Indian ridge system enter the bottom water of the mid-Indian Basin and spread laterally, carrying heat, chemicals, and reduced compounds, including hydrogen sulfide and methane, that provide energy sources for chemosynthetic microbial communities on and above the seafloor. A deep basin with elevated hydrothermal chemical input, low oxygen conditions, warm intermediate water, and a poorly characterized biological community that has been evolving in isolation from the deep-sea faunas of other ocean basins for tens of millions of years since the Indian Ocean reached its current configuration. That is the environmental context for the object in the footage, and every element of that context points in the same direction, toward a biological system that is genuinely distinct from the better-studied deep-sea environments of the Pacific and Atlantic, that has been subject to different selective pressures for a sufficiently long time to have produced genuinely different biological solutions to the problems of deep-sea life, and that contains communities that are not adequately described by the existing literature on deep-sea biology, which is itself primarily based on expeditions to the Pacific and Atlantic. The researchers who are making this argument are not claiming that the distinctive physical oceanography of the Indian Ocean explains what was filmed by the fishing vessel. They are claiming that it provides a physical and biological context in which something genuinely novel, something that does not fit the existing categories of deep-sea life, is more plausible than it would be in a better studied, better oxygenated, more thoroughly characterized deep basin. The Indian Ocean's darkness is not just optical, it is scientific. And what lives in that scientific darkness, shaped by millions of years of evolution in conditions that differ meaningfully from those of the deep oceans we know better, remains genuinely and consequentially unknown. The institutional response to the Sri Lankan fishing vessel footage has followed a trajectory that is by now familiar to anyone who has tracked how scientific and governmental bodies respond to anomalous deep-sea findings that fall outside established categories, and the familiarity of the trajectory does not make it less significant or less worth examining in detail, because the specific way in which institutions respond to anomalous findings tells you something important about the nature of the finding itself, about how seriously the people with access to the full data are taking it, and about the gap between what those people know and what the public is being told. The response has had three distinct phases, each separated from the next by a period of institutional silence that was itself informative, and the current state of the institutional response, which is in the early stages of its third phase, is the clearest indicator yet of how the scientific and governmental communities with access to the footage and the associated data are assessing what the footage shows. The first phase began within 48 hours of the sighting, when the Sri Lankan Marine Research Station where the footage was received formally notified IOTC, the Indian Ocean Tuna Commission, which serves as one of the primary international bodies coordinating marine research and fisheries management in the Indian Ocean, and the Intergovernmental Oceanographic Commission's Indian Ocean Global Ocean Observing System Office, known as IOGOOS, which coordinates the deployment and management of oceanographic monitoring infrastructure across the basin. Both notifications were routine in form, filed through the standard channels that the citizen science program connecting commercial fishermen to the research station had established for reporting unusual sightings. Both notifications produced responses within 72 hours, and both responses were, in retrospect, unusual in their speed and in their content. The IOTC response came from the Commission's science director rather than from the routine notification handling staff, which was itself a departure from the normal procedure, and it requested the full footage and all associated metadata, including the vessel's GPS track, the timestamp records from the camera system, and any physical samples that had been collected from the water in the vicinity of the sighting. The IOGOOS response came from the program coordinator and requested the same materials, adding a request for the contact details of the fishing vessel captain and crew members who had directly observed the object. Both responses arrived within 72 hours on a data set that the research station had not yet formally characterized or analyzed, which means that someone at both institutions had elevated the notification above the routine handling level and had recognized, from the brief description included in the notification, that the footage warranted immediate senior-level attention. That recognition, happening simultaneously at two separate international bodies within 72 hours of a routine citizen science notification, is the first indicator that the footage was being assessed by people who understood its potential significance before the formal scientific analysis had been completed. The research station provided the requested materials to both institutions and received, in return, a request from IOGOOS to treat the footage and associated data as confidential, pending a review by an expert panel that the Commission would convene within 30 days. The research station's director agreed to the confidentiality request, partly because the institutional relationship with IOGOOS was important to the station's funding and operational capacity, and partly because she assessed that the review process would likely accelerate the scientific analysis of the footage by bringing in expertise that the station did not have internally. The 30-day confidentiality period became 60 days, then 90. The deadline extended twice by the expert panel, which communicated to the research station only that its review was ongoing and that additional expertise was being consulted. At the end of the 90-day period, the expert panel issued a summary finding to the research station that described the footage as showing an unidentified biological or possibly biological object of unusual characteristics requiring further investigation, and recommended that the station not release the footage publicly until a follow-up expedition to the target area had been conducted to collect additional data. The summary finding did not describe the footage in any more detail than that. It did not assess the size of the object, did not address the bioluminescence anomaly, did not comment on the historical reports that had by that point been located in the European archives, and did not explain the basis for the characterization of the object as possibly biological rather than simply biological, a qualification that the research station's director noted with attention because the footage, in her assessment, showed something that was unambiguously alive, and the hedging of that assessment by an expert panel that had spent 90 days reviewing the same footage was a signal rather than a clarification. The second phase of the institutional response began when the research station's director, exercising her judgment as an independent scientist, whose institution was not bound beyond the initial 90-day confidentiality period, shared a written description of the footage and the historical reports with 3 colleagues at universities in India, Australia, and France, asking for their informal assessment. All 3 colleagues responded within a week. All 3 assessments expressed significant interest and significant concern, and one of the 3, the Australian colleague, a marine biologist at the University of Western Australia with expertise in deep-sea cephalopod biology, flew to Sri Lanka within 2 weeks of receiving the description to review the footage directly. Her reaction to the footage, described afterward in a message to her department head that has since been shared informally in the research community, was that the footage showed something she could not identify, that the bioluminescence was unlike anything she had seen in 20 years of deep-sea biology, and that the size and behavioral characteristics of the object were inconsistent with any known species in the Indian Ocean deep-sea fauna, or, to her knowledge, in the deep-sea fauna of any other basin. She used a specific phrase in that message that has been repeated in the conversations circulating among the researchers who have been briefed on the sighting, a phrase that captures the quality of the scientific response more precisely than any other formulation that has emerged from those conversations. She wrote that reviewing the footage felt like reading a sentence in a language you almost recognize, where every word is familiar, but the grammar is wrong in a way that makes the meaning impossible to reconstruct. Every element of what she was seeing, the size, the bioluminescence, the behavior, the apparent deep-sea origin, existed within categories she knew, but the combination of those elements in a single organism violated the rules of the biological grammar she had spent her career learning. An organism that is that large and that luminous and that capable of surface presence from abyssal depths is not a sentence that the existing grammar of deep-sea biology can parse. And unparsable sentences, in science as in language, mean one of two things. Either the sentence is wrong, which in this context means the footage is a misidentification or a fabrication, or the grammar needs revision, which means the existing framework of deep-sea biology is missing a category that the footage is revealing. The researchers who have reviewed the footage are not treating it as wrong. They are treating it as a grammar problem. And the work of revising the grammar to accommodate what the footage shows is the work that the third phase of the institutional response is now, slowly and carefully, beginning to organize. The third phase is the one that is currently underway, and it involves the planning of a dedicated deep-sea expedition to the mid-Indian Basin with instruments capable of characterizing the biological and physical environment of the target area at the resolution required to detect and document whatever the fishing vessel filmed. The expedition is being planned by a consortium that includes the Sri Lankan Research Station, the University of Western Australia, a French oceanographic institution with access to a deep-rated research vessel, and a private marine research foundation with autonomous underwater vehicle capacity rated to 6,000 meters. The planning is at an early stage, the funding is not fully secured, and the timeline for the expedition is uncertain. But the fact that 4 institutions from 4 countries are committing resources to planning it is the most concrete institutional signal yet that the footage is being taken seriously at a level that goes beyond polite interest. The expedition, if it proceeds on its current timeline, would reach the target area of the mid-Indian Basin within the next 8 to 14 months, and what it finds there, or does not find, will be the next major data point in a story that has been building in the archives and the logs and the informal conversations of the Indian Ocean research community for considerably longer than anyone currently involved in the investigation had previously realized. The deep-sea biology community's response to the Sri Lankan footage, beyond the institutional framework of expert panels and expedition planning, is happening in the informal channels of scientific communication that precede formal publication and that are, for anyone trying to understand where the scientific consensus on an anomalous finding actually stands, more informative than the official statements that institutions eventually produce. Those informal channels, the emails and conference calls and hallway conversations at scientific meetings, and the comment threads of preprint servers where specialists engage with each other in the technical language of their fields, have been active since the description of the footage began circulating in the research community, and the conversations happening in them are characterized by a quality of engagement that is distinct from the engagement that routine scientific findings produce. Routine findings are discussed in the language of incremental progress, of results that confirm or extend existing understanding, of methods that can be improved and conclusions that can be refined. The conversations about the Sri Lankan footage are happening in a different register, one that the researchers involved are not entirely comfortable with, because it is the register of genuine uncertainty about something fundamental, the register of a field confronting data that its existing frameworks cannot accommodate, and that requires it to ask questions it does not currently have the tools to answer. The deep-sea biologists, who have been most actively engaged in these conversations, are not junior researchers excited by novelty. They are senior scientists with decades of experience in the deep ocean, who have encountered many anomalous findings, and who have developed, through that experience, a finely calibrated sense of which anomalies resolve into known categories on closer examination and which ones do not. Their assessment of the Sri Lankan footage is that it is in the second category, and that assessment is based not on a single anomalous characteristic, but on the combination of characteristics that, taken together, make the footage resistant to explanation within the existing biological framework in a way that none of its individual elements would be if encountered separately. The size anomaly alone would be remarkable, but not unprecedented in the context of deep-sea gigantism, the well-documented tendency of deep-sea organisms to achieve larger body sizes than their shallow water relatives, a phenomenon attributed to the combination of cold temperatures, high pressure, and limited food supply that characterizes the deep ocean and that selects for life history strategies involving slow growth, late maturity, and large adult body size. Deep-sea gigantism is real. It is documented across multiple taxonomic groups, and it provides a framework within which an organism significantly larger than its known relatives could plausibly exist in the deep Indian Ocean fauna. The bioluminescence anomaly alone would be remarkable, but potentially explicable as a novel variant of the luciferin-luciferase chemistry, or as a previously undocumented bioluminescent mechanism in an organism whose biochemistry had not previously been characterized. Novel bioluminescent chemistries have been discovered in the past, and the deep Indian Ocean fauna is sufficiently poorly documented that the existence of organisms with uncharacterized bioluminescent systems is entirely plausible. The behavioral anomaly of surface presence from abyssal depths alone would be remarkable, but potentially explicable as a stress response or a navigational error in an organism whose normal depth range was somewhat shallower than the 4,800 meters of the seafloor below the sighting location, with the apparent vitality of the object being a misreading of footage that does not actually show the behavioral detail required to assess physiological state with confidence. Each of these individual explanations has weaknesses and requires assumptions that the data does not fully support, but each is within the range of the scientifically discussable. What makes the footage genuinely resistant to the existing framework is the combination of all three anomalies in a single object. An organism that is simultaneously in the upper range of what deep-sea gigantism can plausibly produce, producing bioluminescence with spectral characteristics that do not match any known marine bioluminescent chemistry, and demonstrating surface presence from abyssal depths with apparent behavioral agency, is not an organism that can be accommodated by extending any single existing biological framework. It requires the simultaneous extension of multiple frameworks in the same direction toward the existence of something larger, more biochemically novel, and more physiologically capable than anything currently documented in the Indian Ocean deep-sea fauna. The probability of all three frameworks requiring simultaneous extension in the same organism, by chance, without any underlying biological reality connecting them, is low. The more parsimonious interpretation, the one that requires fewer independent assumptions, is that the footage shows a real organism whose biological characteristics are genuinely outside the documented range of known Indian Ocean deep-sea fauna, and that the three anomalies are not independent but are connected properties of a single biological entity whose existence the scientific literature has not previously captured. The most experienced researchers in the conversations are arriving at this interpretation not with excitement but with the specific discomfort of people who understand what it implies for the field. If the footage shows a real organism outside the documented range of known fauna, then the question of why it has not been documented previously is not a trivial one. The Indian Ocean has been crossed by ships for thousands of years, surveyed by oceanographic expeditions for more than a century, and fished by commercial fleets for decades. The historical reports in the archives suggest that the phenomenon has been occurring in the mid-Indian basin at least since the 19th century. An organism of the apparent size and bioluminescent intensity of the object in the footage should not have been completely absent from the scientific record for 150 years if it regularly surfaces in a section of ocean that is crossed by shipping routes and fished by commercial fleets. Its absence from the scientific record requires explanation, and the explanations available are either that it surfaces very rarely, making chance encounters unlikely on any given crossing of the target area, or that it surfaces primarily at night and in conditions of limited visibility, making visual detection difficult even when it does occur, or that it surfaces in a specific geographic area of the mid-Indian basin that is not on the primary shipping routes and is not regularly visited by fishing vessels, making the probability of encounter low even if surfacing events are relatively frequent. The fishing vessel's position at the time of the sighting, 340 kilometers southwest of Sri Lanka, places it at the northern edge of the mid-Indian basin, in water that is deeper than the main fishing grounds but not so remote that commercial vessels never operate there. The vessel was in the area because it was following a tuna aggregation that had moved south of the normal fishing grounds, a displacement that the captain attributed to unusual surface temperature conditions in the normal fishing area. The unusual surface temperature conditions that displace the tuna aggregation are themselves worth examining in the context of the story, because they connect the biological sighting to the physical oceanographic setting of the Indian Ocean in a way that the researchers have identified as potentially significant. The Indian Ocean has been warming faster than any other major ocean basin over the past several decades, with surface temperature increases that are among the most rapid in the global ocean record. That warming is not confined to the surface. It is penetrating into the intermediate and deep layers of the Indian Ocean through the same circulation processes that make the deep Indian Ocean warmer and less oxygenated than other basins. And it is altering the thermal structure of the water column in ways that are affecting the vertical distribution of organisms throughout the basin. Tuna aggregations are moving. Fishing grounds are shifting. And the physical conditions of the deep Indian Ocean, including the temperature and oxygen structure of the abyssal water column above the mid-Indian basin, are changing in ways that could be affecting the biological communities living there in ways that have not yet been documented, because the monitoring infrastructure required to document them does not exist. The displacement of the tuna aggregation that brought the fishing vessel to the northern edge of the mid-Indian basin and the unusual surface temperature conditions that drove that displacement are therefore not simply background context for the sighting. They are a thread connecting the sighting to the broader pattern of Indian Ocean change, and following that thread leads to questions about whether the surfacing of the object in the footage is itself connected to the changing conditions of the deep Indian Ocean, whether whatever drove the object to the surface is related to the same. Physical and chemical changes that are displacing tuna aggregations and altering the thermal structure of the water column, and whether the current period of rapid Indian Ocean warming is producing conditions in the deep basin that are, for reasons that are not yet understood, bringing things to the surface that have not previously surfaced, or surfacing more frequently than they have previously surfaced, or surfacing in locations where they are more likely to be observed than in the historical periods from which the archival reports come. These questions are speculative. They are the kind of questions that arise when you place a biological anomaly in its full physical and oceanographic context and ask whether the context is coincidental or explanatory. The researchers who are asking them are not claiming answers. They are claiming that the questions are worth asking seriously, and that asking them seriously requires the kind of deep Indian Ocean monitoring and research capacity that does not currently exist, and that the current sighting and the historical record it connects to, and the physical oceanographic changes it is embedded in, collectively provide the most compelling argument yet for building. The question of what the object in the footage actually is, in biological terms, has been approached by the researchers involved through a process of systematic elimination that begins with the broadest categories of known deep-sea organisms and works progressively toward the smaller set of possibilities that remain after the less plausible candidates have been removed. And the process of elimination is itself informative because of how many candidates it removes and how few it leaves. The elimination process begins with the vertebrates, because the size of the object places it within the size range of the largest known marine vertebrates, and the vertebrate deep-sea fauna, while poorly documented in the Indian Ocean, is the best documented component of the deep-sea biological community globally. The largest deep-sea fish known from the Indian Ocean and from the global deep-sea fauna generally are the oarfish, Regalicus glesni, which can reach documented lengths of up to 8 meters and which has a strongly bioluminescent appearance due to its highly reflective silvery scales, and the giant oarfish has historically been invoked as a possible explanation for sea serpent reports from mariners in multiple ocean basins. The oarfish is eliminated as a candidate for the Sri Lankan footage on multiple grounds. Its maximum documented length of 8 meters falls well short of the 20 to 28 meter size estimate from the footage. Its bioluminescence is passive, a reflection of ambient light from its scales rather than active light production, and would not produce the self-illumination visible against the background of the vessel's floodlights that the footage shows. And its behavior at the surface is well documented from the numerous stranded and distressed specimens that have been encountered over the years, and it is not consistent with the volitional descent described in the footage. The oarfish is the most plausible vertebrate candidate, and it is not plausible. Moving through the other large deep-sea vertebrates, the sleeper sharks of the family Somniosidae, which include the Greenland shark and the Pacific sleeper shark, and which are known to occur at abyssal depths in some ocean basins, are eliminated on the grounds that no known shark species produces bioluminescence of the intensity and spectral characteristics described in the footage, and that the largest known sleeper sharks reach maximum lengths of approximately 7 meters, well below the size estimate. The beaked whales, which are among the deepest diving marine mammals, and which have been documented at depths exceeding 3,000 meters in some species, are eliminated on the grounds that they are air-breathing mammals that must return to the surface regularly, and whose surface behavior is well documented and entirely inconsistent with the characteristics of the object in the footage. The entire vertebrate category is eliminated by this analysis, not because large unknown vertebrates are biologically impossible in the deep Indian Ocean, but because no known vertebrate group has members whose combination of size, bioluminescence, and behavioral characteristics is consistent with what the footage shows, and the extension of known vertebrate biology required to accommodate the footage. Characteristics is too large to be supported by the available evidence without additional confirmation. The invertebrate candidates require more detailed analysis because the deep-sea invertebrate fauna is more diverse, more poorly documented, and includes groups with a wider range of body plans, physiological capabilities, and bioluminescent strategies than the vertebrates. The cephalopods, the group that includes squid, octopus, and cuttlefish, are the most obvious invertebrate candidates because they include the largest known marine invertebrates, the giant and colossal squid, because they are well represented in the deep Indian Ocean fauna, even though that fauna is poorly documented, and because cephalopods are among the most sophisticated bioluminescent organisms in the deep ocean, with complex photophore systems capable of producing patterned light displays for communication and camouflage. The giant squid, Architeuthis dux, is the largest known cephalopod at approximately 13 meters maximum documented length, and the colossal squid, Mesonychoteuthis hamiltoni, reaches approximately 14 meters. Both fall short of the size estimate from the footage. Both produce bioluminescence in the blue-green range rather than the white-yellow range described in the footage, and both are known, from the few living specimens that have been observed, to behave in ways inconsistent with the sustained surface presence and volitional descent described in the footage, showing instead the passive floating or distressed behavior typical of organisms that have ascended beyond their pressure tolerance. The known giant cephalopods are eliminated as candidates on similar grounds as the vertebrates. They are the most plausible candidates within their taxonomic group, and they are not plausible. What remains after the elimination of the known vertebrate and cephalopod candidates is a set of possibilities that are progressively less well characterized and progressively more speculative. And it is in this residual set that the researchers are now focusing their analytical attention. The first residual possibility is an unknown species within a known taxonomic group, specifically an unknown giant cephalopod species in the Indian Ocean deep-sea fauna that exceeds the documented size of Architeuthis or Mesonychoteuthis, and that has evolved a bioluminescent chemistry different from the blue-green range of known cephalopod bioluminescence. This possibility is consistent with the known pattern of deep Indian Ocean biological novelty, and with the general principle that the largest organisms in poorly documented faunas are the most likely to have escaped formal scientific description. An unknown cephalopod of 20 to 28 meters with white-yellow bioluminescence and enhanced pressure tolerance, allowing surface presence from abyssal depths, would be a remarkable discovery. But it would be a discovery within a known biological category rather than a category-level novelty. It would require the revision of the documented size range of giant cephalopods, the addition of a novel bioluminescent chemistry to the cephalopod repertoire, and the documentation of pressure tolerance capabilities exceeding those of any known cephalopod. But none of these revisions would require the invention of a new biological category. This is the most conservative possibility remaining after the elimination process, and it is the one that the researchers are most comfortable discussing publicly because it stays within the boundaries of known biology, even while extending them significantly. The second residual possibility is more category-level in its implications. The deep ocean contains several phyla of organisms whose representatives at abyssal depths in the Indian Ocean are essentially undocumented, including the ctenophores, the gelatinous comb jellies that are among the most widespread and most bioluminescent organisms in the deep ocean, and the siphonophores, the colonial hydrozoans that include the longest known organisms on Earth, and that produce some of the most sophisticated bioluminescent displays in the marine environment. A siphonophore colony of 20 to 28 meters is within the documented size range of the group, the largest known siphonophore exceeding 40 meters, and a siphonophore with novel bioluminescent chemistry would be consistent with the poorly documented state of Indian Ocean deep siphonophore biology. The behavioral characteristics of the object in the footage, specifically the volitional descent, are the primary challenge for the siphonophore hypothesis, because siphonophores are generally not capable of the kind of directed, controlled vertical movement that the footage appears to show. But the deep Indian Ocean siphonophore fauna include species that have never been studied in living condition, and the behavioral capabilities of organisms that have been known only from preserved specimens or brief footage encounters are not well constrained. The third residual possibility is the one that the researchers discuss least publicly and most carefully, the one that sits at the boundary between extending known biology and requiring genuinely new biological categories. It is the possibility that the object in the footage represents an organism from a deep-sea lineage that has been evolving in the isolation of the deep Indian Ocean for long enough, and under selective pressures different enough from those operating in other deep-sea environments, to have produced a body plan and a set of physiological capabilities that do not correspond to any known phylum-level category in the existing biological literature. The deep ocean has produced biological novelties at the phylum level before. The phylum Loricifera, microscopic animals living in marine sediments, was not discovered until 1983. The phylum Cycliophora, tiny organisms living on the lips of Norway lobsters, was not discovered until 1995. These are small organisms, and the likelihood of a phylum-level novelty at the scale of 20 to 28 meters is lower than at microscopic scales. But the deep Indian Ocean abyssal fauna is sufficiently poorly characterized that categorical statements about what phylum-level novelties it could or could not contain are not supportable by the available data. The researchers are not claiming that the footage shows a new phylum. They are claiming that the footage shows something they cannot identify within the existing framework, that the elimination process has removed the most plausible known candidates, and that the residual possibilities range from a large, unknown species within a known group to something more categorically novel, and that determining which of those possibilities is correct requires the kind of direct physical investigation of the target area that the planned expedition is intended to provide. The discomfort with the third possibility is not scientific. It is institutional and cultural. The discomfort of a field that has learned from long experience that extraordinary claims require extraordinary evidence, and that the evidence currently available for the Sri Lankan footage, while compelling, is not yet at the level that would support a claim of categorical biological novelty in the scientific literature. The footage is real. The historical record is real. The physical oceanographic context is real. The elimination of known candidates is real. What is not yet real, in the sense of being supported by evidence at the level required for formal scientific claim-making, is the conclusion that the elimination process leads toward. That conclusion is coming. The expedition will either confirm it or refute it. And the researchers who have done the elimination analysis are, in the careful private language of people who have followed the evidence to an uncomfortable place and who understand what it means if they are right, not expecting to be refuted. The climate dimension of the Sri Lankan sighting is the thread that connects this story from the biology of a single observed object to the broader condition of the Indian Ocean as a system under stress, and it is the thread that transforms the sighting from a remarkable biological anomaly into something with implications that extend beyond taxonomy and species discovery into the functioning of an entire ocean basin that is changing faster than any monitoring system currently deployed in it is equipped to detect. The Indian Ocean has been warming at a rate that exceeds the warming rate of any other major ocean basin over the instrumental record, and the consequences of that warming are visible in the surface ocean through changes in sea surface temperature, shifts in monsoon dynamics, alterations in the distribution and abundance of commercially important fish species, and an increase in the frequency and intensity of marine heat waves that have caused large-scale coral bleaching events across the tropical Indian Ocean reef systems. These surface consequences are documented, monitored, and increasingly well understood because the surface of the Indian Ocean is accessible to the satellite and ship-based monitoring infrastructure that has been built up over the past several decades. What is less documented, less monitored, and less well understood is the consequence of that surface warming for the deeper layers of the Indian Ocean, for the water column below the thermocline where the biological communities of the mesopelagic, bathypelagic, and abyssal zones live, and where the physical and chemical conditions that those communities depend on are changing on timescales and through mechanisms that the available monitoring infrastructure was not designed to capture. The warming of the Indian Ocean surface is not confined to the surface. Heat is being transported into the subsurface ocean through a combination of processes, including the deepening of the mixed layer, the increase in the temperature of the water masses being subducted below the thermocline, and the modification of the intermediate and deep water mass properties by the changing surface conditions at their formation sites in the Southern Ocean. The Argo float network provides some visibility into the subsurface warming through its temperature profiles down to 2,000 meters, and the Argo data for the Indian Ocean shows a pattern of subsurface warming that is consistent with the downward penetration of the surface heat anomaly into the intermediate layers of the basin. But below 2,000 meters, where the Argo floats do not go, the thermal state of the Indian Ocean deep water column is known only from the sparse and infrequent ship-based hydrographic surveys that have sampled those depths on timescales of years to decades rather than the continuous monitoring that the upper ocean receives. The deep Indian Ocean is warming, based on the available evidence, but the rate and the spatial distribution of that warming are poorly constrained, and the consequences for the biological communities living in those depths are essentially unmonitored. The connection between the warming deep Indian Ocean and the Sri Lankan sighting is not direct in the sense of a simple causal chain, but it is real in the sense that the physical conditions of the deep Indian Ocean are changing, and changing conditions in any biological system produce biological responses, and biological responses in a system as poorly monitored as the deep Indian Ocean are likely to manifest as anomalies before they manifest as understood processes, because the baseline against which they would be recognized as changes does not exist with sufficient resolution to detect anything short of a dramatic departure from the expected state. The object filmed by the fishing vessel camera is a dramatic departure from the expected state of the surface Indian Ocean at 4 in the morning, and if it is connected in any way to changing conditions in the deep basin, that connection is not something the available monitoring infrastructure is capable of establishing, because the monitoring infrastructure was not built to detect biological responses to deep ocean warming in the abyssal Indian Ocean. The oxygen dimension of the Indian Ocean warming adds a layer of biological concern that is independent of the temperature change itself, but that interacts with it in ways that are relevant to the biological communities of the deep basin. Warming seawater holds less dissolved oxygen because the solubility of gases in water decreases as temperature increases. As the Indian Ocean warms, the oxygen content of its water is declining, a process called ocean deoxygenation that is occurring globally, but that is particularly significant in the Indian Ocean because of its already low baseline oxygen concentrations in the deep water column. The oxygen minimum zone of the Indian Ocean, already among the most severe in the global ocean, is expanding both vertically and horizontally as warming reduces oxygen solubility and as increased surface productivity. In some regions, increases the oxygen demand of microbial respiration in the subsurface layers below. The expansion of the oxygen minimum zone is compressing the habitable depth range of aerobic organisms in the Indian Ocean water column, squeezing the mesopelagic community into a narrower vertical band between the oxygen-poor surface of the minimum zone and the depth at which temperature becomes limiting for their physiology. This compression has been documented in several regions of the Indian Ocean through analysis of acoustic backscatter data from research vessels. which shows changes in the depth distribution of the deep scattering layer consistent with organisms adjusting their vertical range in response to the changing oxygen structure of the water column. Organisms are moving, their depth distributions are shifting. The vertical ecology of the Indian Ocean mesopelagic is reorganizing in response to the changing physical and chemical environment, and the direction of that reorganization towards shallower depths for organisms being pushed upward by expanding low-oxygen water below is relevant to the question of what might drive a deep-sea organism to the surface. If the oxygen minimum zone is expanding upward and the habitable depth range for aerobic deep-sea organisms is being compressed from below by anoxic conditions and from above by the thermal and photochemical conditions of the surface layer, then organisms that have historically lived within a comfortable depth range may be finding that range contracting, and the behavioral and physiological responses to that contraction may include ascent to depths that the organisms would not normally inhabit, including, in extreme cases, the surface. This is a speculative connection. The object in the footage, if it is a deep abyssal organism from 4,800 meters, lives far below the oxygen minimum zone that is affecting the mesopelagic community, and the oxygen minimum zone expansion, while significant, has not penetrated to abyssal depths in the Indian Ocean. But the principle that changing physical and chemical conditions in the Indian Ocean water column are driving biological responses that include vertical redistribution of organisms is documented at mesopelagic depths, and the question of whether analogous processes are operating at deeper levels, driving biological responses in the bathypelagic and abyssal communities that might include unusual vertical movements, is not answerable with the available monitoring data because the monitoring data does not exist for those depths. The Indian Ocean Biological Monitoring Gap is the deepest and most consequential monitoring gap in the global ocean observation system, and the Sri Lankan sighting is the most dramatic single piece of evidence for the significance of that gap that has emerged in the past decade. It is dramatic not because it proves that something terrible is happening in the deep Indian Ocean, but because it demonstrates in the most concrete possible way that the deep Indian Ocean is capable of producing phenomena of the apparent scale and biological novelty of the object in the footage without any existing monitoring system having detected the conditions that produced it, without any existing biological framework having predicted its existence, and without any existing institutional response mechanism having been prepared for the information management challenges that its appearance has created. The gap between what the Indian Ocean is doing in its deep layers and what the surface world knows it is doing is not a gap that can be closed by better interpretation of existing data. It is a gap that requires new infrastructure, new expeditions, new monitoring systems, and a fundamental reorientation of the priority given to deep Indian Ocean research relative to the other components of the global ocean observation system. The planned expedition to the mid-Indian Basin is a step toward closing that gap, but it is a small step relative to the scale of the gap, and the timeline of 8 to 14 months for the expedition to reach the target area is a timeline during which the deep Indian Ocean will continue doing whatever it is doing, unmonitored at the resolution required to understand it, producing phenomena that the existing frameworks cannot predict or explain, and occasionally, apparently, sending something to the surface that makes a fishing vessel captain pick up the phone at 4 in the morning and describe, in the only language available to him, what it looks like when the ocean decides to show you something it was keeping. That sentence, from a man with 20 years of ocean experience who was not speaking metaphorically, is the most honest description available of what the Sri Lankan sighting represents at the level of the relationship between the surface world and the deep ocean. The ocean has been keeping things. The keeping has been enabled by the monitoring gap, the biological darkness, the institutional inertia, and the assumption of unremarkability that has characterized the scientific community's approach to the deep Indian Ocean for as long as systematic ocean science has existed. The things being kept are not static. They are living, and they are responding to a changing ocean in ways that are not yet understood, and they are, at least occasionally, coming to the surface. What comes next, in the deep Indian Ocean and in the scientific investigation of what the fishing vessel camera recorded, will be determined by whether the surface world treats the appearance of something it cannot explain as a reason to look more carefully at the system that produced it, or as an anomaly to be managed through confidentiality agreements and expert panels and the slow machinery of institutional response until the next sighting makes the management strategy untenable. The ocean is not waiting for the management strategy to be resolved. It is doing what it is doing, in the dark, at depth, in the warming, deoxygenating water column of the least monitored major ocean basin on Earth. And the next thing it decides to show us may not arrive at 4 in the morning alongside a single fishing vessel in the southwestern Indian Ocean. It may arrive somewhere more visible, in conditions less amenable to the careful information management that has characterized the response to the current sighting, and in a form that makes the question of what lies in the deep Indian Ocean impossible to defer to a planned expedition that is still 8 to 14 months from sailing. The fishing vessel that recorded the footage in the southwestern Indian Ocean has returned to its home port in Sri Lanka and resumed its normal operations, and the captain who made the 4 in the morning phone call to the Marine Research Station has given one informal interview to a journalist working for a regional maritime publication, an interview in which he described the experience in terms that have stayed with everyone who has read them, and that capture something about the nature of the encounter that the scientific analysis, for all its rigor and precision, has not fully managed to convey. He said that in 20 years of working the Indian Ocean, he had developed a feeling for the sea, a bodily sense of the ocean as a known and navigable environment whose moods and surprises fell within a range that experience had taught him to anticipate and manage. He said that the 40 minutes during which the object was alongside his vessel was the only time in his professional life that the feeling had left him entirely, replaced by something he described not as fear exactly, but as the specific vertigo of realizing that the environment you thought you knew had a dimension you had not accounted for, a depth that was not merely physical, but categorical, a darkness that was not merely optical, but ontological. He was not speaking as a scientist. He was speaking as a man who had spent 20 years building a working relationship with a body of water, and who had encountered, in a single pre-dawn hour, evidence that the relationship was less complete than he had believed. That incompleteness, the gap between what the ocean presents at its surface and what it contains in its depths, is the central truth of this story, and it is a truth that the scientific record, the biological analysis, the physical oceanographic context, the historical archive, and the institutional response all converge on from different directions without any of them fully exhausting it. The deep Indian Ocean is not known. It is assumed, mapped at low resolution, sampled at sparse intervals, monitored inadequately, governed by frameworks designed for a different ocean and a different era, and populated by biological communities that the existing scientific literature has barely begun to describe. The object in the fishing vessel footage is the most dramatic single piece of evidence for that incompleteness that has emerged in recent memory, but it is not the only evidence, and it is not the most important evidence. The most important evidence is the totality of what the deep Indian Ocean monitoring infrastructure does not show. The vast, dark space in the data record where the abyssal biology, the deep circulation, the hydrothermal chemistry, and the biological responses to a warming and deoxygenating water column should be visible and are not, because the instruments to make them visible have not been deployed, and the expeditions to characterize them have not been funded, and the institutional priority that would drive both has not been established. The Sri Lankan sighting changes that calculus, or it should. It provides a concrete, documented, multi-institutional, historically grounded case that the deep Indian Ocean contains phenomena of sufficient scale and biological novelty to justify the level of investment and attention that it has never previously received. And it provides that case in a form that is more compelling than any theoretical argument about monitoring gaps or biological incompleteness, because it arrives not as an abstract statement about what might be in the deep ocean, but as footage of something that was at the surface of the Indian Ocean at 4 in the morning and that descended back into the darkness before anyone with the appropriate instruments could reach it. The planned expedition to the mid-Indian Basin is the scientific community's response to that evidence, and it is the right response, and it is insufficient. It is insufficient not because the expedition is poorly designed or inadequately resourced for what it is, but because what the situation requires is not a single expedition, but a sustained, comprehensive, multi-year, internationally coordinated program of deep Indian Ocean research that is commensurate with the scale of the monitoring gap and the potential significance of what the gap is hiding. A single expedition, however well equipped, can survey a fraction of the target area at a snapshot in time. It can collect biological samples and seafloor imagery and water column chemistry data from the specific locations its autonomous vehicles visit, and it can answer some of the questions that the fishing vessel footage has raised. But it cannot characterize the full biological community of the mid-Indian Basin. It cannot establish the baseline monitoring record that would allow changes in that community to be detected and understood. It cannot determine whether the object in the footage is a resident of the target area or a visitor, whether its surfacing was a unique event or a recurrent behavior, whether it is a single species or a colonial organism, whether its biochemistry represents a novel bioluminescent system or a known one in an unknown configuration. These questions require the kind of sustained engagement with the deep Indian Ocean that no single expedition can provide, and they require it urgently because the deep Indian Ocean is not static. It is changing, warming, deoxygenating, reorganizing its biological communities in ways that are unmonitored at depth, and that may be producing phenomena of which the Sri Lankan sighting is the most visible, but not the only expression. The urgency is real, and it is grounded not in alarm about the specific object in the footage, but in the understanding that a changing ocean produces changing biology, and changing biology in a system as large and as consequential as the Indian Ocean has consequences for the surface world that extend from the stability of fisheries to the efficiency of the biological carbon pump to the chemistry of the atmosphere above the basin. Those consequences are not hypothetical. They are the normal outputs of a biological system responding to physical and chemical forcing, and the deep Indian Ocean is receiving more physical and chemical forcing than at any point in the recent geological record, and its biological response to that forcing is essentially unmonitored. The object in the footage is a symptom. The disease, if that word is appropriate for what is happening to the deep Indian Ocean under the combined pressures of warming, deoxygenation, and the accumulated consequences of a century of industrial fishing and maritime activity, is the broader transformation of a biological system that we do not understand well enough to know what we are losing or what we are producing as we change the conditions under which it operates. What the marine biologists who have seen the footage are terrified of is not the object itself. It is what the object implies about the depth of their ignorance, about the scale of the biological system that exists below the surface of the Indian Ocean in conditions they have not adequately characterized, about the changes that may be occurring in that system in response to a warming and deoxygenating ocean, and about the consequences of those changes for a surface world that depends on the Indian Ocean's biological and chemical functioning in ways that are not fully understood, but that are certainly significant. The terror is the terror of competent people confronting the limits of their competence in a situation where the stakes of that limitation are high, and the time available to close the knowledge gap may be shorter than the institutional machinery required to close it can operate. It is a rational terror, grounded in evidence, calibrated to uncertainty, and pointing toward a specific and actionable response. More monitoring, more expeditions, more resources, more international coordination, more honest public communication about what the deep Indian Ocean contains and what it is doing and why it matters to every person who lives on the surface of a planet that is 71% covered by water, whose deepest layers remain, in the most consequential possible sense, unknown. The footage from the fishing vessel camera will eventually be released. The scientific community will eventually publish its analysis. The expedition will eventually sail, and it will eventually return with data, and that data will either confirm the most remarkable interpretation of the footage, or it will provide a more conventional explanation that the current evidence has not made available. Either outcome will be significant. A conventional explanation would close one anomaly while leaving the broader monitoring gap intact and the historical record unresolved. A confirmation of something genuinely novel would open a scientific and public conversation about the deep Indian Ocean that is long overdue, and that the current monitoring infrastructure is not equipped to support at the scale that conversation would require. The ocean is waiting, as it always waits, indifferent to the timelines of the institutions that are supposed to be watching it, doing what it does in the darkness below the thermocline, in the cold and pressure of the abyssal zone, in the warming and deoxygenating water column of the least monitored major ocean basin on Earth. Something surfaced from the deepest part of the Indian Ocean. It was there for 40 minutes, lit from within by light that no known marine organism produces, larger than any known invertebrate, alive in conditions that should have made survival impossible, and then it descended back into the darkness from which it came. The darkness is still there. The monitoring gap is still there. The warming is still there. The biological communities of the deep Indian Ocean are still there, doing whatever they do in conditions we have not adequately characterized, responding to changes we are not adequately tracking, producing phenomena we are not adequately equipped to detect or explain. This channel will be here when the expedition returns. It will be here when the footage is released. It will be here when the preprints are posted and the peer review is completed, and the institutional response evolves beyond confidentiality requests and expert panels into the kind of sustained scientific engagement that the deep Indian Ocean has always deserved and never received. Subscribe and stay close, because the Indian Ocean is not finished showing us what it has been keeping, and the next thing it shows us may arrive with less warning and in conditions less amenable to the careful, patient, evidence-based investigation that this story has been built on. The deep ocean is the last frontier on this planet. It is changing. It is alive in ways we do not fully understand. And it is occasionally sending things to the surface. Pay attention to what surfaces. It is trying to tell us something about the darkness below.